So I'd just like to thank Roger for organising this amazing meeting and for inviting me to speak today. And he, of course, along with his father, created the intellectual foundations for much of what we'll talk about today. And I was quite delighted, actually, that in the previous session, they mentioned Yuri Lavsblik's paper on Can a Biologist Fix a Radio, which is quite helpful for me, because this idea of the human body as a machine metaphor, which began, of course, with René Descartes in 1641 and was developed by Hobbes and Julien de la Mettrie, and then David Hume, who in 1779 talked about a stupid mechanic who designed the human body and other biological systems. And that actually is the key problem, is that we're not designed particularly well, and we don't have an instruction manual. So thank you for that, giving me that context. So in 1620, Francis Bacon, and then in 1627, after he died, wrote a book, Novum Organum and then New Atlantis, in which he stated that the true task of science is not to catalogue things, but actually to uncover the generative rules that build those things. He called those the secret motions of things. And if you could do that, if you could unpick those generative rules of life, which is what they were, you could enlarge the bounds of the human empire, in his own words. And really, in modern day terms, really what we're talking about here is what I would call the generative grammar of life. And what I'd like to suggest to you today is actually that we've reached a critical moment in the history of humankind, and indeed of all life on Earth. And really, one can think about this as really a phase transition. And I don't think life on Earth can or ever will be the same again. So for the last 4 billion years, all life on Earth has evolved by a Darwinian and Wallacean process of evolution by natural selection, described by them together in 1858 in London. And the key point about Darwinian evolution by natural selection is that it's blind, it lacks foresight, it lacks intentionality, and most importantly, it lacks design. It has no notion of the future, it's purposeless. Now, what's changed? What is the paradigm shift? What's changed is that we now are at the beginning of what I would call the age of AI-driven biological design, where we can use genome language models with incredible architectures like striped hyena and superlative ability to broaden the context windows and define attention to the nucleotide level. We can combine and converge those technologies with the ability to construct DNA molecules at scale, all the way up to genomes. And this is work that my colleagues at Stanford and Caltech have been doing. We've kind of put them all together in a single home, and the convergence of the ability to use AI to design genomes and the ability of new technologies to build them is really immensely powerful and opens up what I would call this new age of genome writing. And you could call it generative biology. Some people call it engineering biology. But what we have to realize is this is fundamentally different to the world that we used to live in, when we received nature and we could tinker with it by breeding, we could edit it in the same way that nature edits, through the corruption of genomes, through mutations and rearrangement. But we couldn't fundamentally reimagine life from first principles. And the problem with that is we get stuck on the historical tree of life, when actually there's this huge DNA sequence space out there, an infinite universe of possibility, but evolution can't efficiently explore that space. It's stuck on this infinitesimally small region. And so really, we're now in what I would call a post-Darwinian age, when there are now two authors, co-authors, if you like, of life. One, of course, is evolution by natural selection, but the other is, of course, human-made design and construction. And these two types of life will coexist necessarily in the future, and it's for us to decide how that happens. So what are we doing? Well, we're turning life, essentially, into a programmable language, much like the computer software that you use every day when you run a computer program. And I see the phase we're at now in human history or technological history is being very similar to the 19th century, when Bessemer reduced the cost of steel production, changing it from an artisanal material used only very sparingly because it was so expensive, into what basically became a technology with geopolitical consequences when it was developed by Carnegie to build railways and infrastructure and bridges and so on. So I'm argue really that biology is set to become infrastructure like steel, and the forces driving that are the ability to turn biology into predictive engineering material through AI-mediated design, and the ability to build DNA rapidly, efficiently, accurately, and at very reduced cost. That's an essential part of it. And it just turns out that in the same way that mathematics was the perfect language to describe physics, that AI seems to be the perfect language to describe biology. And we've, of course, at the gene level, seen some incredibly remarkable results with programs like AlphaFold and DragonFold and Rosetta, which enable us to look at, interrogate databases of structures and define rules or predictive rules without really understanding how it happens, and that's an important point. And then more recently, my colleague Brian He at Stanford, the ARC Institute, has actually designed the world's first AI-driven designed synthetic species. It was a Phi-X virus with only 93% identity to the parental virus, which technically meets the definition of a new species. So obviously, we can't today design more complex organisms, and a virus isn't really a living organism, it's a biological entity. But we've taken off, we're like the Wright brothers kind of bumping into the air. But the point is we've begun, this has happened, and we need to call the moment, and we need to align, this is a civilizational moment, we need to align internationally and within our nation states to ensure that this dual-use technology is used wisely, sensibly, responsibly, ethically, in a moral manner, in a transparent manner, in an equitable manner, and in a way that generally benefits society. So just a quick point to say, how is this different to the CRISPR revolution and gene editing? Well, it's totally different. It's the difference is basically the difference between redlining a document that you're editing, where you're constrained by the text that's on the page, and the ability to have an empty page and just be able to use your imagination to write whatever you like. We can all become Dostoevskys of biology eventually. Right now we're writing children's books, simple children's books, because we only understand the grammar of life at the most rudimentary level. But the fact that we can design viruses indicates to me that this is ahead of us. We need to unpick these grammatical rules, and if we can do that, we can open up, obviously an incredible understanding of biology with huge implications, including what will become a multi-trillion-dollar bioeconomy with implications from things like information storage to biocomputing, to healthcare, biomolecules, biomaterials, reprogramming the genomes of crops, and addressing some of these existential crises like food production and climate change by creating a sustainable technology where you can actually grow things. And I quite honestly predict that we may eventually grow cell phones and grow houses. We'll literally use biology for almost everything you can think of. And in the same way that William Philip yesterday talked about the coming of a second quantum revolution, I believe that we're now at the beginning of what I would call a genomic revolution, in which we can now construct biological systems from first principles. And at this point, we change the classic Baconian paradigm of breaking life down to understand it, to actually constructing life to understand it. It's a huge difference. And Omar Yashi said yesterday that at the heart of science, we make new chemical structures. And now, instead of just making molecules, we're going to be making organisms at the genome level. To do this, however, we need to understand how the instruction manual that we never received, we have to kind of reverse engineer it. And to do that, we need to go beyond the simple linear sequences of DNA and try and understand the higher order structures of DNA, which is where I believe in the 3D genome, the true lost grammar, the rules of the missing grammar, if you like, reside. And if we can unpick those rules, we'll be able to assemble the complete grammar book of genomes encoding particular species. So today we're fortunate to have some wonderful speakers who are going to address various different aspects of the grammar of life and how the grammar goes wrong and how bits of grammar can be detected diagnostically. Unfortunately, David Klenneman wasn't able to join us. It's worth mentioning that David's relevance to this field was obviously his development of next-generation sequencing at Solexa, which then became Illumina. And what that did is it made the grammar of life legible. We could build databases of sequences which we could then interrogate with AI. But the first speaker today is Caroline Luger, who was awarded the World Laureate Association Award in 2023. She's a distinguished professor in biochemistry at the University of Colorado, Boulder. She's done seminal work on the nucleosome, understanding its three-dimensional structure, the way that DNA wraps around the histone core, and also has done some groundbreaking work on Kaposi sarcoma and the way in which viral infections sort of leverage the three-dimensional genome. So I will now hand over to Carolyn, and who's actually going to talk about the physical basis of the grammar of life.